For weeks, we've been talking about the different types of SARS CoV-2 testing, specifically molecular testing and antigen testing. On this episode of Shareable Science Beyond the Blog, we're going to dive a little bit deeper into each type and explore the circumstances where one might be preferable to the other. Welcome to Shareable Science. Science you can share. All right, let's jump right in. These two types of tests to diagnose the presence of the SARS CoV-2 virus, the molecular test, which is also sometimes called the PCR or RT-PCR test, and the antigen test, which is sometimes called the lateral flow assay or lateral flow immunoassay. These tests detect the presence of the virus in different ways. The molecular test is actually looking for the presence of the genetic material, the genome of the virus. The antigen test is actually detecting the presence of proteins that sit often on the outside of the virus, like the spike protein. So these are looking at different ways of detection. And those different ways of detection are the result, or they drive all the differences that we're going to see on the rest of this table. The molecular test actually can be done with a number of different samples. Those deep nasal swabs, the swabs just inside the nostril, saliva, and actually other tests from deeper in the respiratory system. Antigen tests only focus on the deep nasal swabs or the swabs just inside the nostril. They have not yet been approved for other samples. They could be at some point, but right now they're not. All right, for the rest of these, it's important to note that the molecular test actually requires the sample to be analyzed inside a specialized machine in a lab by a technician. So it takes a day or more, and if there's a lot of backup, it can take several days to get a result. But that extra step of using that specialized equipment means it's incredibly sensitive. You can pick up on the order of probably a few thousand copies of the virus genome with this kind of approach. It requires specialized equipment, so it can't necessarily just be done in a doctor's office or in a walk-in clinic, and certainly not at your house. And the cost is also expensive because of the equipment and the technician time. So $35 to $200 per test. You can reduce that some if you do pooling, which we've talked about in a previous episode, but it's still pricey. But again, high specificity. It's very, very sensitive. All right, the antigen test, 15 to 30 minutes. That's a huge difference. But the trade-off there for that quick result is it's less sensitive. You have to have a much higher amount of virus present. Not a few thousand samples, a few thousand copies of the genome, but you probably have to have tens of thousands or hundreds of thousands of copies of the virus in order for you to test positive on an antigen test. Generally, it doesn't require any specialized equipment, so it can be done at a point of care. And there are a number of people that are trying to develop tests that you could do at home. And then much less expensive, $1 to $5 per cost on average. Okay, so with that in mind, let's talk about the different ways that these tests might get used. And that requires us talking about the difference between diagnostic testing. So I have someone who has got symptoms and I want to know if they are infected. Or they're asymptomatic, but they've had an exposure and I want to know if they have the virus. Versus group screening, where I'm taking a group of people that I don't know anything about them. I don't know that they've had an exposure. I'm asking, can this group of people, can I bring them back to school? Can I bring them back to work? Can I bring them back for a social setting? I'm essentially, with group screening, trying to identify individuals who are infectious and remove them from my population to slow the spread. That's different from I need to clinically know if someone is infected. So with that distinction in mind, molecular tests do this really, really well. This is what they are designed to do, to identify who is infectious from a clinical setting. There are challenges when you try to bring that into a group screening because of the delay in results, because of the increased cost, and some other pieces that we're going to talk about. So potentially, the rapid antigen testing may be a better approach for this. Now, let's walk through this graph. There's a lot going on here, so hang with me. Let's talk about what's on the axis of our graph. So on the x-axis, I've got the days since you've been exposed. So this is the time course since you were exposed at day 0. On the y-axis, I've got the amount of virus, the viral load that's in your system. And you're going to see that this is a log base 10. So every time I move up a number, I am increasing by a factor of 10. So 2, I'm talking about hundreds of copies. 3, I would be talking about thousands of copies. 4, I'd be talking about tens of thousands of copies. And then this black line shows you generally the amount of virus, the increase in the amount of virus and the slow drop in the amount of virus over a patient's course of infection. Everybody's going to have a slightly different shape for this. Some people's are going to be higher, some are going to be lower, some are going to be longer, some are going to be shorter. But in general, you get the point that there's this really rapid increase. While the virus has the upper hand, it's increasing exponentially. And then as your body's defenses begin to catch up and it begins to turn the tide, there's this slow decline. Now, there's a window here where you have a high enough level of virus in your system that you are infecting other people. And that's what we really want to pay attention to as we think about detecting whether somebody is infected and removing them from a population so they don't infect other individuals. Now, I've got 2 dotted lines here, a green line and a blue line. The dotted green line shows you the level of detection, the sensitivity of that molecular, that PCR-based test. Remember, I told you it's on the order of thousands of samples. The blue line shows you the sensitivity limit, the lower limit for the amount of virus from an antigen test. So you can see it's 100-fold higher. So it does a poorer job of detecting low amounts of virus, but it does a good job of detecting large amounts of virus. So the argument is that with screening, I might be doing these antigen tests, these cheap rapid antigen tests every day or every other day, and I would be detecting the window where someone is infectious, as opposed to a PCR test where I may only be doing this once a week. And I may detect infection, but I might actually also miss that window. So that's really the crux of the argument. Does it make more sense to be looking at something that is admittedly less sensitive, but to be testing more frequently, or to be looking at that higher sensitivity test? And a number of folks are arguing that this higher sensitivity test should be used only for diagnostic, and that we should be seeing more and more of the lower sensitivity for group screening. Now, before I let you go, because I know we've covered a lot, let's talk just a second about false positives and false negatives. No test is perfect 100% of the time. All tests are going to have instances where an individual has a positive test, but they aren't actually infectious, or an individual has a negative test and they are infectious. There are more false positives and false negatives among the antigen tests than among the PCR-based tests. And so that's a ding against them. The proponents of the frequent antigen tests argue that if you're doing this every day or every other day and you get a false negative, so you're really infectious even though the test says negative, if you're doing this again the next day, you will catch it at that point, and you're still going to pull somebody out before there's been days and days of being infectious and being out in the public. If you have a false positive, so you don't really have the infection, but the test registers a positive, many groups are recommending that you do one of 2 things. One, you have a standby different antigen test that maybe isn't looking for the spike protein, but it's looking for a membrane protein. And so when you test positive for the spike protein, you follow it up with an antigen test for the membrane protein. And if you are truly positive, you would test positive in both cases. The other argument is that you follow up a positive antigen test with a molecular test. So you're using now that diagnostic setting. Lots of details here, I know. And we don't right now have large amounts of antigen tests that we could think about doing these at home on a daily basis. They haven't been approved by the FDA for that use. So we've got a ways to go. This argument and this discussion is still taking shape, but we're hearing more and more about these rapid antigen tests being used as a screening, and I wanted you to have that background. I hope you find this useful, even if it's a bit convoluted. Please feel free to share this with those around you, especially that might be interested in different types of testing and the difference between diagnostic and screening. Thank you for watching. I look forward to seeing you on the next episode of Shareable Science Beyond the Blog. Take care.